Warning, your DNA just issued a delete command. Yes, your cells come with a built-in delete command. It's called apoptosis, and it's how your body cleans house. One protein flip, and a cell slices its own DNA, packs itself into tiny garbage bags, and disappears. Now, what if modern frequencies, the stuff from phones, Wi-Fi, chargers, nudge that button more often? Too much delete, and tissues thin and fail. Too little, and damaged cells stick around and turn rogue. The big question tonight, are today's signals quietly steering DNA toward a suicide script? We'll test that claim with real lab data, the P53-inch judge, oxidative sparks, and what actually helps. Here's where we're going. First, we'll meet the delete command. What apoptosis is, why evolution installed it, and how it actually runs in a cell. Then we'll look at how cells listen to tiny signals, chemical sparks, ion flows, redox shifts, and why small nudges can trigger big pathways. After that, we'll map the frequency soup we live in. Power lines, Wi-Fi, 4G per 5G, Bluetooth, chargers, all with simple ranges and how fast energy drops with distance. Then we'll cross the bridge where this story lives. Oxidative stress. That's where reactive oxygen species, ROS, can flip from helpful messages to harmful noise that pushes cells toward delete. From there, we go to evidence. In vitro cell studies, animal work, and human data, you'll hear the mainstream stance. Non-ionizing radio frequency doesn't have enough energy to break DNA directly. You'll also see papers reporting increased RAWs, calcium shifts, or apoptosis markers under certain exposures. We'll separate what's been replicated from what hasn't, and we'll call out limits like heating confounds and cell line quirks. We'll also give the guardian of the genome his scene, P53 deciding repair or remove. Then we'll stress test the claim with dose, distance, modulation, and the weird window effects where biology reacts at specific ranges. Finally, we land on practical steps that lower baseline stress without panic or snake oil. Rules of the game. Facts first, hype never. Ionizing versus non-ionizing matters. If a study is disputed, we say so. If a mechanism is a theory, we label it. Our method is simple. Cause. Signal, pathway, outcome. If that chain breaks anywhere, we don't sell it. Cool? All right, load the delete command. The delete command, Apoptosis 101. Imagine a city that fixes itself by quietly removing broken buildings before they collapse. That's your body. Apoptosis is the permit system and the demolition crew. Evolution put it there to shape you in the womb, prune your immune cells, and keep would-be cancers from taking off. Without it, fingers fuse, immune cells attack the wrong targets, and damaged cells linger until they cause bigger problems. With too much of it, you lose neurons, muscle, and organ function, so the body keeps it tight. Here's how the delete runs. Inside each cell, there's a set of proteases called caspases. Think of them as scissors that only cut when someone removes the safety cap. The intrinsic pathway starts at the mitochondria, the cell's power stations, and weirdly also its executioners. When a cell senses serious trouble, like heavy DNA damage, major oxidative stress, or messed up proteins, the outer mitochondrial membrane gets more permeable. That's controlled by the BCA2 family, the bouncers at the door. Some members are pro-def, like BAX and BAC. Others are anti-def, like BCA2 and BCA-XL. When the pro-def side wins, mitochondria leak cytochrome C. That's the signal that assembles the apoptosome, which activates caspase 9, which then flips on executioner caspases 3 and 7. Those chopped structural proteins cleave a break called ICAD and unleash CAD, the nuclease that ladders DNA into neat fragments. It's disturbingly tidy. There's also the extrinsic pathway. That's when a cell gets a kill order from outside. Def receptors like Fas or TNF on the membrane bind their ligands. That pulls together disc complexes, which activate caspase 8, and the scissors party starts. In many cells, both routes meet up at the mitochondria, because caspase 8 can activate BID, which tips Bax back toward pore formation. The routes are different doors to the same end. Who decides which door opens? Enter p53, often called the guardian of the genome. It's a transcription factor that wakes up when DNA is hurt. Double strand breaks, weird replication stress, or oxidative lesions. Stabilized p53 pauses the cell cycle and buys time for repair. If the damage is fixable, repair teams patch it, and p53 stands down. If not, p53 tilts the BCL2 balance by upregulating pro-death proteins like PUMA and NOXA, and the mitochondria open up. So, in a very real way, p53 is the judge that reads the file and says repair or delete. Now, what trips the alarms in the first place? Plenty of normal stuff. Cells constantly deal with reactive oxygen species. They're not just bad, they're signaled. A short pulse of ROS can nudge pathways to adapt, grow, or defend. But if ROS builds up beyond what antioxidants like SOD, catalase, and glutathione can buffer, it damages lipids, proteins, and DNA bases. Markers like 8-OEGG pop up, and damage sensors engage. Misfolded proteins, ER stress, calcium overload, and persistent inflammation all feed into the same decision tree. The point is, the delete command isn't random. It listens to context. This is important for our central question. If chemistry speaks the language of charges, electrons, and ions, then physical fields might whisper in that same language. Membranes have voltages. Channels open and close with tiny changes in electric potential. Calcium influx can set off huge cascades. Mitochondria run on an electrochemical gradient. So in theory, very small physical nudges could, under the right conditions, move a cell from fix it to end it. That doesn't mean every nudge does. It means the system is responsive by design. There's also the Goldilocks idea, called hormesis. Small, short stress can be useful. It trains the response systems like exercise does for muscles. Big or chronic stress breaks things. With apoptosis, a modest redox pulse might help clear out the most damaged cells and keep a tissue healthy. Too much redox chaos or the wrong cells at the wrong time, think neurons, and you get degeneration. That's why the stakes here cut both ways. Too little delete invites tumors. Too much delete thins tissues and brains. Let's zoom into the details the body uses to keep this precise. The caspase cascade is nonlinear. Once a threshold is crossed, it goes fast, which is why timing matters. Mitochondrial membrane potential, basically how charged that inner membrane is, can wobble when a cell is stressed. A drop there makes cytochrome C release more likely. BCL2 family proteins respond to dozens of upstream cues. DNA damage markers like ATM, ATR, oxidative shifts, cytokines, and calcium spikes. Def receptors are kept in check until ligands arrive from immune cells that have vetted a target. It's layers of brakes and gas pedals, not a single on-off switch. And then there's cleanup. Apoptotic cells flip phosphatidylserine to the outer surface and eat me flag. Neighboring cells and macrophages recognize it and swallow the debris quietly. No mess, no inflammation, no alarms to the whole tissue. That's a huge difference from necrosis, which spills guts and triggers inflammation. Your body prefers quiet deletions. So yes, there is a delete key. It exists to keep you alive, not to betray you. It sits inside a network that checks damage, balances signals, and acts when the cost of repair is higher than the cost of removal. Our job now is to ask a sober question. Can modern signals, non-ionizing fields from everyday tech, meaningfully tilt the inputs to that decision? Not by smashing bonds like X-rays do, but by nudging the redox and calcium dials that mitochondria and p53 read. We'll get there. First, a quick look at how cells act like antennas, how tiny pushes become big choices. Because if a whisper can change a verdict, we should know how the courtroom hears. Signals in cells, how little pushes make big changes. Have you ever wondered why a tiny spark can start a whole fireworks show inside a cell? Cells are electrical, not just kind of. The membrane around each cell holds a voltage, like a charged battery. On one side, more positive ions, on the other, more negative. That separation is energy, and it runs the gates. Proteins that open and close to let ions like sodium, potassium, and especially calcium slip through. A few thousand ions move, and suddenly enzymes wake up, genes switch on, mitochondria speed up or slow down, small push, big ripple. Calcium is the star of this whisper becomes a shout story. Resting calcium inside a cell is tiny, nanomolar low. A brief pulse all by itself can flip whole pathways. It binds to Ca-modulin, which flips on kinases like CAMKI. That changes which proteins get phosphorylated, which changes what they do. It also talks to the nucleus. Transcription factors respond. And now the cell is making different tools for the next hour, the next day. And because calcium also hits mitochondria, it tunes energy output. And if it floods, leans towards stress and even the apoptotic machinery. That's why researchers care so much about anything that might nudge calcium channels, especially the voltage-gated ones on the membrane. Now let's talk redox, the chemistry of electrons and oxygen. We throw raws around like it's a villain, but honestly, it's more like a text message. Low, short pulses of reactive oxygen species carry info. They tweak cysteine switches on proteins, modulate signaling, and prime defenses. Antioxidants aren't there to erase all raws. They're there to keep the message short and clear. When production outruns clean up, I think too many texts at 3 a.m., the messages turn into noise. Lipids oxidize, proteins misfold, DNA bases take hits, and stress alarms trip. Markers like 8, OHG tell you the messages went off the rails. So how do tiny pushes get past the membrane? Mechanosensitive and voltage-gated channels feel changes in stretch and charge. Receptors bind ligands and start cascades that amplify with every step. One receptor activates 10G proteins, which activate 100 enzymes, which make thousands of second messengers. That's built-in gain. It's why biology can react to whispers without cooking itself. And it's why non-thermal stimuli, signals that don't heat tissue measurably, are still a scientific question. If the gate responds to a charge change, you don't need a furnace, you need the right nudge at the right time. This is where windows come in. Cells don't respond linearly to everything. Sometimes there's a sweet spot. Too weak and nothing happens. Too strong and systems clamp down. Hormesis lives here. Small stress sharpens responses. Exercise is a perfect example. A bit of damage, a bit of ROS, then the rebound makes you stronger. But keep pushing past recovery and you get injury, not growth. Cells use the same trick. Short redox pulses train antioxidant defenses. Chronic overload depletes them. And now the same cue that used to help flips toward harm. Let's make it concrete. Voltage-gated calcium channels open with small changes in membrane potential. If a stimulus, chemical or physical, leans the voltage a few millivolts toward opening, you get more calcium spikes. That elevates mitochondrial work, which makes more RAWs as a byproduct. If antioxidant systems match that rise, you adapt and get tougher. If they don't, RAWs lingers, tweaks P53 stabilization, and the damage versus repair math shifts. Same initial nudge, different context, very different outcome. That's the knife edge the cell walks every day. Mitochondria sit at the pivot. They read calcium and redox in real time and adjust ATP output. But their inner membrane potential, the charge across it, is fragile. A drop makes it easier to release cytochrome C when other signals say, it's time. That doesn't mean a little wobble equals doom. It means the system sums inputs. DNA damage signals from ATM, ATR, cytokine noise from neighbors, calcium pulses, Ross tone. The final vote decides whether to pause and fix or call in the caspases. Could it be that physical fields nudge the same levers chemistry does? That's the open question. We know cells are antennas for ions in charge. We know small pattern inputs can flip big pathways. We also know most safety standards focus on heating, not on narrow windows where non-thermal nudges might matter. So we test, we look for consistent cause, signal, pathway, outcome chains. No chain, no claim. Next, let's map the frequencies actually around us and how strong they are so we can see which nudges are even plausible in real life. The frequency soup we swim in. Let's map the noise we actually live in so we're not arguing with ghosts. Start at the slow end. Power. The stuff in your walls hums at 50 or 60 hertz. Big motors, power lines, induction cooktops, all in the extremely low frequency, ELF, band, wavelengths the size of cities, fields that rise when current flows, and they fall fast with distance. A step back matters. Slide up to kilohertz and megahertz. Old school AM and FM sit here, plus some baby monitors and walkie-talkies. Then the modern daily drivers, radio frequency and microwave. Wi-Fi runs at 2.4 and 5 gigahertz, sometimes 6 in newer routers. Bluetooth hangs out near 2.4 as well, but uses tiny bursts and low power. Your phone does 4G around 700 megahertz up to a few gigahertz depending on the band, and 5G adds more lanes. Sub 6 gigahertz like 3.5, and in some cities, millimeter wave up around 24 to 39 gigahertz for short hops. Now, strength, picture a flashlight. Close up, it's bright. Move away, it fades fast. That's the inverse square law in practice. Double the distance, quarter the power density. Phones near your head, higher local exposure. Phone on a table, big drop. Routers across the room, even bigger drop. Chargers and wireless pads, they use near-field coupling at lower frequencies for a few centimeters. Beyond that, they fall off a cliff. That's why distance remains the cheapest shield on Earth. Near field versus far field matters. Within about a wavelength or two of an antenna, fields can be messy. Electric and magnetic parts don't travel together yet. They wrap around the source. Past that, they lock into a wave and spread predictably. At 2.4 gigahertz, a wavelength is roughly 12.5 centimeters. So your body is usually in the far field for room-to-room Wi-Fi, but in the near field for a phone pressed to your ear or a watch on your wrist. Tiny changes in where you hold a device can change exposure more than any sticker ever will. Let's clear the big physics line. Ionizing versus non-ionizing. Ionizing radiation, UV, X-rays, gamma, carries enough punch per photon to rip electrons off molecules, break bonds directly, and write damage into DNA on contact. That's not what phones and Wi-Fi emit. Radio frequency and microwave bands are non-ionizing. A single photon that doesn't have the energy to snap a bond. The established boring mechanism at these frequencies is heating. Energy absorbed by tissue turns into a tiny temperature rise. That's why safety standards like IC-NIRP and IEEE base limits on specific absorption rate, SAR, and tissue heating thresholds with big safety margins. So why the debate? Because biology isn't just a bag of water. Cells run on charges and timers. Some studies report effects at levels below significant heating, shifts in calcium handling, reactive oxygen species, or stress proteins, while many others don't. That's the non-thermal effects conversation. Standards today say protect against heating, and you're good. Critics say, what if certain patterns, pulses, modulations, duty cycles, matter more than average power? Think of it like sun through blinds. Same sunlight, different flicker on your eyes. Let's talk numbers without drowning. A phone by your head can hit a SAR up to 1 to 2 watts per kilogram in a small region. But that's tested under worst case continuous transmission. Real calls hop and rest. A Wi-Fi router across the room, power densities in the microwatts per square meter range for most homes, which is orders of magnitude below heating thresholds. Millimeter wave 5G, it doesn't dive deep. Energy is mostly absorbed in the first millimeters of skin. That changes which cells see it. Skin, nerve endings, immune sentinels, while deeper tissues see much less. Environment adds layers. In a car, metal can reflect and concentrate signals. In dense apartments, you see many networks, but each contributes a sliver and distance still wins. Body-worn gear lives in your near field, so watch bands and earbuds are closer, but run at very low powers with duty-cycled whispers. The net effect is a patchwork. Spikes when you stream, and the phone ramps up, lulls when it idles. So the soup is real, but it isn't a bathtub of power. It's a mist with hot spots near sources. And the key variables you control, distance, time, and placement, beat most of the fancy gear you see online. Now that we've got the map, we can ask a fair question. Where in this landscape could signals plausibly nudge redox or calcium enough to matter? And when does the math say no way? That bridge runs straight through mitochondria and oxidative stress. Let's cross it. Oxidative stress and the mitochondrial pivot. Let's step onto the bridge where this whole story either holds or falls apart. Oxidative stress. Mitochondria sit at the center like a switchboard. They make ATP, the cell's energy coin, by moving electrons down the respiratory chain. Most of the time, that chain is tidy. A tiny fraction leaks and forms reactive oxygen species, superoxide, hydrogen peroxide. At low levels, those molecules are signals. They tune metabolism, trigger defenses, help cells adapt, push that production higher, or let cleanup lag, and the same signals turn into static that trips alarms. Here's the pivot. Mitochondria don't just make energy. They're wired into the delete decision. Their inner membrane holds a steep voltage, the proton motive force. When workloads spike or calcium floods in, the voltage can sag. If it sags far enough, and pro-def proteins are already lining the outer membrane, you get pores. Cytochrome C slips out. That's the point of no return. The moment the caspase cascade lights up. So anything that shifts redox tone or calcium handling can lean on that decision. How do we know redox crosses the line? We measure markers. 8 OGG flags oxidize DNA bases. Lipid peroxidation products like MDA or 4-HONE say membranes took hits. Enzymes like superoxide dismutase and catalase rise or fall depending on stress and training. In healthy adaptation, you'll see a brief Ross pulse, then an antioxidant bump that overshoots and resets balance. In chronic overload, the markers stack up and defenses flatten. That's when P53 stabilizes longer, cell cycle pauses stretch, and the repair or remove math shifts. Now, the voltage-gated calcium channel idea. It's a theory that non-ionizing fields at certain patterns might nudge membrane sensors, so a few extra calcium channels open. Not heating, not bond breaking, a small bias on an already twitchy gate. More calcium spikes mean mitochondria work harder, which can increase electron leak and ROS as a byproduct. If the antioxidant systems are robust, well-slept, well-fed, not inflamed, those bumps probably get soaked up. If you're stacked with stress, poorly slept, high inflammation, that same bump could land differently. This is why timing and pattern matter. Biology doesn't run on averages, it runs on pulses. Calcium comes in spikes, not smooth ramps. Raw signaling is bursty by design. Cells react to the on-off rhythm. So a pulsed external input could in principle be louder to a cell than a steady input with the same total energy. That's the non-thermal, non-linear window conversation. It doesn't claim magic. It claims that sensors have thresholds and time constants, and if you hit those, you get a bigger response than power alone would predict. Let's ground that with the mitochondrial membrane potential. Picture it like a tight dam holding back protons. Short calcium pulses make turbines spin faster and electrons move quicker. If the dam is sturdy, all good. If the wall is thin from prior hits, oxidized lipids, less cardiolipin integrity, it's easier for pores to form whenever backs and back get the nod from upstream signals. That's the sensitive context idea. Same nudge, different baseline, different outcome. What about direct DNA damage? Non-ionizing fields don't have photon energy to snap covalent bonds. Mainstream standards sit on that bedrock and focus on heating. But DNA can be hurt indirectly when ROS outpaces repair. That's the oxidative route. Base lesions form, repair kicks in, BER, NER, all the alphabet soup. If damage is modest, repair wins, and P53 goes home. If damage keeps coming, P53 stays active, leans on PUMA and NOXA, and mitochondria open up. The bridge is not RF breaks DNA. The bridge is RF could, in specific windows, add to oxidative load or calcium entry, and that load, if unbuffered, tilts fate. We should also talk adaptation because it cuts both ways. Mild redox nudges can harden cells. The antioxidant network scales up. Mitochondria get more efficient. That's hormesis again. A tiny challenge now can protect you later against a bigger hit, say UV on skin. Some studies even report neutral or protective effects under certain 5G, like exposures when paired with another stressor, hinting that context and sequencing matter. It's not that fields heal, it's that biology loves practice reps. Key uncertainties live here. Do everyday exposure patterns create calcium or ROS changes large enough, long enough in real tissues to be biologically meaningful? Lab setups can box a cell line in a uniform field at set SAR and modulation for hours. Real life is messy. Duty cycles, reflections, distance jumps, your phone idling most of the day. That doesn't erase risk. It sets the bar for what counts as a plausible pathway in humans. So, what would a convincing chain look like? Step 1. Define an exposure with careful dosimetry and thermal controls. Step 2. Show a reproducible change in a proximal signal. Calcium spikes, mitochondrial potential wobble, early ROS pulse. Step 3. Link that to downstream stress markers. 8. OGG, lipid peroxidation. SOD shifts, without heat artifacts. Step 4. Demonstrate that the change tips P53 stabilization and BCA2 family expression. Step 5. See caspase activation and apoptotic morphology. Then ideally, step 6. Show the same pattern in an animal tissue and find echoes in human biomarker studies under realistic use. That's a high bar on purpose. Some papers hit pieces of it, some find nothing. That split is the debate. Our job next is to walk through what the lab says. Cell cultures in animals, where signals are easiest to control. If the bridge holds there, we can ask if it ever shows up in people outside the dish. What the lab says, cell and animal evidence. So what actually happens when you put cells or animals in front of these signals and watch closely? Let's start in the dish, because that's where you can control almost everything. In vitro, researchers expose human or animal cell lines to define radiofrequency fields with set SARs, durations, and modulations, while keeping temperature clamped so heating doesn't cheat. Many groups report no change in oxidative stress or DNA repair. A recent study on human skin cells at 3.5 gigahertz using 5G, like modulation up to 4 watts per kilogram, found no rise in reactive oxygen species, no impairment of UV damage repair, and no adaptive preconditioning. Others see the opposite under different setups. Some cell lines, neuronal progenitors, immune cells, and especially sperm, show increased ROS or apoptosis markers after radiofrequency exposure. You'll see caspase 3 activation, a bump in DNA fragmentation by common assays, or phosphatidylserine flipping on the membrane. Sperm cells come up a lot because they're metabolically active, have limited antioxidant reserves, and pack DNA densely. In boar sperm, in vitro 5G, band exposures increase the fraction with damaged DNA in a frequency-dependent way. That's not humans, but it flags a sensitive cell type. Now, apoptosis markers can be noisy. DNA damage in comet assays can reflect transient strand breaks during repair, not permanent harm. That's why better studies pair assays. RAS probes, antioxidant enzyme activity, 8-OGG for base lesions, caspase activity, and morphology under a microscope. When those move together without a temperature rise, you've got a stronger signal. When only one jiggles, you hold the champagne. Modulation and duty cycle matter in the lab. Some teams report that pulsed or amplitude modulated signals push biology more than continuous waves at the same average power. The idea lines up with the pulse-sensitive nature of calcium and raw signaling. But replication is spotty. One lymphocyte study across UMTS channels saw small DNA damage by comet at certain frequencies, yet no rise in ROS, no apoptosis, and no p53 mutations, suggesting a mild, possibly reversible effect rather than a march to cell death. Thermal artifacts are the classic trap. Even a 1-3 degrees Celsius drift in a tiny culture can goose metabolism and ROS. Good rigs use feedback thermometry in the medium, sham exposures with identical hardware and blind analysis. When those guardrails are tight, many effects fade. When they're loose, positive results climb. That's not conspiracy, it's measurement. Switch to animals. Here you get a whole organism with circulation, immune cells, hormones, sleep cycles, the context humans actually live in. Rodent studies show mixed outcomes. Some report oxidative stress markers rising in brain, testis, and liver after weeks of RF exposure. Higher lipid peroxidation, altered superoxide dismutase or catalase, and sometimes increased apoptosis in sensitive tissues. Reproductive endpoints show up again. Sperm count or motility dips, more DNA fragmentation. Other groups, using similar or even higher exposure metrics, find little to no change. Exposure pattern is a big axis here. Intermittent versus continuous fields can yield different biology. A few minutes on, a few off, for hours a day, over weeks, seems to provoke more changes in some studies than a steady field at the same average dose. That fits the pulse response idea. But again, replication is uneven, and strain, age, and baseline health of animals swing results. Specific absorption rate ranges matter. Animal work often runs at SARs, clustered around 4 to 4 watts per kilogram for localized tissues, pushing near safety limits to see if anything moves. In some millimeter wave studies, power is deposited mainly in skin. Results there tend to show skin-level changes, heat shock proteins, immune cell shifts, more than deep organ effects, which is expected given shallow penetration. Let's call out a bright spot for nuance. Some experiments report that pre-exposure to a 5G-like field did not worsen, and in a few cases slightly mitigated damage from a second hit like UV in skin models. That smells like hormesis, mild stress training defenses, rather than any magic protection. It's not a health claim. It's a reminder that sequence and baseline state shape outcomes. Limitations? Plenty. Cell lines adapt to plastic and lab life. They may be twitchier or duller than your tissues. Animals aren't people. Dosing doesn't map 1-to-1. Blinding and sham controls aren't universal. Publication bias favors something happened. And dosimetry, the art of knowing exactly what field the biology saw, is hard. Many older papers don't meet today's dosimetry standards, muddying meta-analyses. So what threads can we pull with some confidence? First, non-ionizing RF at realistic SARs doesn't consistently cause direct DNA breaks in mammalian cells. Second, certain cell types, sperm, neurons, some immune cells, look more sensitive to oxidative shifts under some exposure patterns. Third, markers of oxidative stress and Occasionally, apoptosis can rise in vitro and in animal tissues, but replication is inconsistent and often fades under stricter controls. Fourth, modulation and intermittency might matter more than average power, aligning with the biology of pulses. That points to a cautious bridge. If RF fields do anything in living systems below heating thresholds, the most plausible route is a small nudge to calcium handling or mitochondrial ROS that in stress contexts can tip signaling toward repair delay or rarely apoptosis. That's a far cry from mass cellular suicide, but it isn't nothing forever either. The next question is obvious. Do we see echoes of this in people using phones and Wi-Fi in the wild? Let's go there. Human data, epidemiology, symptoms, and big debates. Okay, dishes and rats are useful. But what happens to people who live around this stuff for years? Let's start with the big picture. Epidemiology. These are pattern spotting studies that ask, do folks with higher exposure see more disease? The classic ones looked at people living near power lines or transmitters, workers around strong ELF fields, and later heavy mobile phone users. For extremely low frequency fields from power lines, some early studies found a small bump in childhood leukemia risk with higher measured magnetic fields. Later analyses pulled that association toward maybe small with lots of confounders. It's still debated, but if there's an effect, it's weak and limited to rare exposure situations. That's not radio frequency though. Different band, different mechanism. For radio frequency, phones, base stations, Wi-Fi, research split into two lanes. One, proximity studies. Do people near cell towers get more cancer? Most well-done studies don't show consistent increases. Power drops fast with distance, and typical tower exposures are far below phone-to-head levels. 2. Phone cohorts and case control studies. Do heavy callers get more brain tumors? Here's where it gets spicy. The Interphone study, a giant multi-country case control project, mostly found no clear increase in glioma or meningioma with regular phone use. There was a hint of higher glioma risk in the top decile of reported use. But recall bias is a beast. Ask someone with a brain tumor to estimate past phone time, and you get fog and overestimates. Later, Scandinavian registry studies tracking actual tumor rates over decades of rising phone adoption didn't show a matching surge. That weighs against a big population-level effect. Still, in 2011, the WHO's IARC classified RF fields as possibly carcinogenic to humans, Group 2B. That's the same bucket as pickled vegetables and coffee used to be in limited evidence in humans, less than sufficient in animals. It wasn't a verdict of harm, it was a flag for more research. Since then, tech changed, 3G to 4G to early 5G, so exposure patterns shifted while our long-term data clock keeps ticking. What about day-to-day symptoms? People report headaches, sleep trouble, brain fog, skin tingles around devices. This cluster often gets labeled electromagnetic hypersensitivity. Double-blind provocation studies bring folks into a lab, turn fields on and off without telling them, and measure symptoms, usually don't find a reliable ability to detect exposure better than chance. That points to a strong nocebo component. Expectation drives symptoms, but that doesn't mean folks aren't suffering. It means the trigger might be more about stress, sleep, and attention than fields alone. Physiology studies try to split the hair. Some track heart rate variability, EEG changes, or sleep architecture with and without phone-like exposures. Results are mixed. Tiny shifts here and there, often within normal variability, and replication is shaky. If there's an effect, it's small and probably swamped by coffee, light at night, or doom scrolling before bed. Let's be real. Your screen content is likely doing more to your sleep than the radio inside the phone. Now, biomarkers. A handful of human studies look at oxidative stress markers. 8, OEGG in urine, lipid peroxidation products, antioxidant enzyme levels. Before and after defined exposures, like a few hours of phone use or over longer intervals in workers. Some report modest increases after acute heavy use. Others see no change. When studies tighten controls, standardize diets, and monitor actual emitted power, effects often shrink. One careful human skin cell study in vitro at a 5G relevant 3.5 gigahertz found no oxidative stress and no hit to DNA repair up to 4 watts per kilogram. In humans, millimeter wave exposures mostly hit skin. Limited field studies so far don't show consistent systemic oxidative shifts. So where's the big debate? It sits on 3 stools. First, mechanism. Mainstream bodies hold that non-ionizing RF doesn't have enough energy per photon to break DNA, so they base safety on heating. Critics argue that biology is pulse sensitive and that voltage-gated channels in mitochondria could amplify weak pattern inputs into redox or calcium changes. Second, consistency. Many lab positives fail to replicate under stricter dosimetry. Critics say windows and modulation aren't matched. Defenders say artifacts vanish with better controls. Third, relevance. Some animal studies show oxidative stress in tumors under high chronic exposures. Translating that to human everyday use is messy. Here's a useful angle. Dose and distance in real life. Your biggest RF dose is still your own phone near your body. Tower exposure is usually far lower. That means if epidemiology were to see anything, it would likely correlate with heavy, long-term, close proximity phone use. Not just living in a city with 5G. Yet tumor registries haven't shown a dramatic shift. If there's a risk, it's probably small, focused on certain tumor types, and hard to separate from diagnostic drift and better imaging. Where do agencies land today? WHO and national health bodies say current evidence doesn't confirm health risk below guideline limits, but they advise simple precaution. Use speaker mode, limit kids' direct head exposure, and avoid carrying an active phone in a pocket all day. That's not fear, it's low-cost sense. So human data doesn't scream delete key pressed, but it also doesn't close the book. The strongest human signal so far lives in edge cases. Very heavy, long-term users and sensitive endpoints like male fertility, where oxidative stress could plausibly play a role. Which brings us back to the cell's judge. If ROS ticks up even a little, does P53 notice? Let's meet the guardian who reads those signals and decides repair or remove. The judge, P53 and friends decide fate. Meet the bouncer who can shut the whole club down, P53. When things are calm, P53 is kept low and quiet. A chaperone called MDM2 tags it for disposal, like taking out the trash on schedule. But the second DNA damage shows up. Double-strand breaks lighting up ATM and ATR kinases, or base lesions that stall replication. Those kinases slap phosphate tags on P53 and MDM2. Translation: stop the trash pickup. P53 levels rise fast. Now the judge is on the bench. First move isn't death, it's a timeout. P53 orders a cell cycle pause by turning on P21, which blocks cyclin-dependent kinases. That buys minutes to hours for repair crews. NER homologous recombination to patch the mess. If repair works, P53 steps down, MDM2 resumes the cleanup, and the cell goes back to work. No drama, just maintenance. If the scan still looks ugly, P53 changes tone, it starts writing death warrants, it turns up pro-apoptotic genes like PUMA and NAGA, tips the BCL2 family toward BAX and BAC, and primes the mitochondrial pore. It can also push FAS and DR5 on the surface, making the cell more responsive to outside "you're done" signals. That's the dual role. Repair chief first, executioner only when the math is bad. Where does ROS fit? Reactive oxygen species are both evidence and amplifier. Oxidative-based damage stabilizes P53 by activating ATM-ATR. P53, in turn, can increase ROS by dialing down antioxidant programs or upregulating pro-oxidant enzymes in a last-ditch push to expose weak cells. Or, in mild stress, it can do the opposite, support antioxidant genes and help recovery. Context matters. Low, brief ROS pulses nudge p53 toward repair. Sustained ROS holds p53 high and nudges apoptosis. So could non-ionizing fields lean on this lever? The plausible path isn't RF snaps DNA. It's RF, under certain pulse patterns and doses, slightly boosts calcium entry or mitochondrial workload, which raises ROS. If that bump is small and your baseline is good, sleep, diet, low inflammation, p53 likely rides it out as a training rep. If you're already stacked with hits, UV, toxins, chronic inflammation, that same bump might push p53 to stay stabilized longer. That could mean longer pauses, or in delicate tissues, a few more cells crossing the apoptotic line. That's a theory, not a verdict. Lab data are mixed. Some studies see no oxidative change at 5G, like exposures even up to 4 watts per kilogram in skin cells. Others, in sensitive cells like sperm, report more DNA fragmentation and apoptosis markers under certain frequencies. Different tissues, different thresholds. Let's not forget p53's friends. Upstream, ATM, and ATR sense different flavors of DNA trouble, blunt breaks versus stalled forks, and feed into p53. Downstream, BAX and BAC are the door openers on mitochondria. p53 also has a cytosolic side hustle. It can bind BCL and BCL2 directly and free BAX faster, a shortcut when time's up. Meanwhile, the NF-kB pathway can pull the other way, promoting survival and inflammation, which complicates the readout. And the unfolded protein response in the ER talks to p53 too. Chronic misfolded proteins push chop and can prime apoptosis. All these paths cross at the same gate. When p53 fails, stakes flip. If p53 is muted by mutation, common in many cancers, damaged cells keep dividing. Tumor risk rises because the judge can't call delete anymore. If p53 is hyperactive in the wrong context, you get tissue loss and degenerative changes. That's why the system is tuned. You want p53 decisive, not trigger happy. The EMF angle lands here. If fields shift ROS a hair, do we see p53 stabilization? A tight in vitro skin study at 3.5 gigahertz said no under their settings. Other work in different cells suggests yes under specific patterns. Duty cycle and modulation could matter more than raw power, because biological sensors read pulses. That's why replication is hard. Change the pulse, the distance, or the temperature control, and biology might switch from ignore to notice. So what's our honest call on the judge? p53 is built to weigh lots of small signals and make a binary choice. Modern frequencies at everyday levels are unlikely to shove p53 into constant delete mode across healthy tissues. But in edge cases, high local dose, sensitive cells, stacked stress, you could see small shifts in the repair versus remove balance through the oxidative route. That's not a panic headline. It's a nudge hypothesis that matches the best fit mechanisms and the mixed lab record. To test that fairly, we have to talk dose, distance, and those weird biological windows where small pulsed inputs loom larger than averages. Because if p53 is reading the rhythm, not just the volume, then proximity and pattern become the real story. Let's stress test the claim with numbers you can actually control. Dose, distance, and the window problem. Let's stress test this with numbers you can actually use. Start with the simplest rule in physics land. Inverse square. Double the distance from a small antenna, quarter the power you absorb. That's why a phone on your desk is a very different exposure than a phone on your ear. It's not subtle, it's orders of magnitude. In most homes, far-field Wi-Fi levels sit way below heating thresholds, and distance makes them fall even more. So if there's any meaningful biological nudge, it's going to happen up close for longer and likely in pulses. Now the window problem. Biology doesn't always scale neatly with dose. You can have 3 zones, too weak to matter, a narrow window where sensors notice, and high levels where systems clamp down or you hit plain heating. Voltage-gated calcium channels don't care about your average. They care about brief voltage tips that cross an opening threshold. Same with redox pulses. A short, patterned burst can ring louder than a steady whisper, even at the same total energy. So you get odd curves. Nothing, then a response, then flat again. Duty cycle is the sleeper variable. Your phone doesn't shout non-stop. It chirps, listens, rests. Streaming ramps power, idle drops it. A smartwatch pings in short packets. 2 exposures with the same average power can feel very different to a cell if one is chopped into fast on-off pulses that land on calcium's timing sweet spots. That's why some lab hits show up with amplitude modulated or pulse signals and vanish with a pure continuous wave. Proximity multiplies all of this. Near field is messy, and your body can act like an antenna piece. Pocket carry? You've put a small transmitter right against tissue for hours, with bursts every time an app checks in. Flip to speaker or bag carry, and you just bought a 10x to 100x reduction without thinking. That's the cheapest lever you have, and it's boringly effective. Cumulative time matters in a human way, not a lab way. A cell dish gets 3 hours steady. You get 10 minutes here, 2 minutes there, 8 hours of a phone sleeping on a nightstand. Sum that into 2 buckets, high proximity bursts, and low-level background. The background is almost certainly under any plausible non-thermal window for most tissues. The bursts are where you look for edge case nudges, especially in sensitive cells like sperm in a front pocket or skin under a millimeter wave handset. Let's sanity check with realistic profiles. Phone at ear for a 15-minute call twice a day, otherwise on a desk. Router across the room. Laptop on a table, not your lap. That exposure map puts the highest SAR near your head for half an hour total, with big distance everywhere else. Registry data hasn't shown a tumor wave matching decades of this pattern, which weighs against large effects. But for fertility, multiple studies, human, observational, and animal, flag pocket carry and heat as risks. Radio plus warmth plus low antioxidant reserve is the trifecta you can avoid with one habit change. Here's where standards and windows collide. IC-NIRP in IEEE limits anchor to heating with big safety margins. They don't model every possible non-thermal window because those windows, if they exist, vary by tissue, timing, and modulation. So the practical stance is twofold. Stay under limits and reduce close-in time-heavy exposures where biology is most likely to hear pulses. If a window sits anywhere relevant to daily life, you'll hit it less often with distance and fewer bursts. What about meters and shields? Measuring far field in a room can help you map hotspots. But remember, your phone is the main source you control. Stick-on chips claiming to harmonize fields don't change SAR. Cases that detune antennas can force your phone to work harder, raising power. That's backwards. If you buy anything, buy a longer cable for your charger, so your nightstand isn't a docking bay, and maybe a headset you actually like, so you use it. And pattern still isn't destiny. Baseline biology decides how a nudge lands. Sleep debt, chronic inflammation, poor diet, those shrink your safe zone and make small stresses stack. Fix those and tiny inputs get swallowed by robust buffering. That's hormesis doing its job. So the verdict on dose and distance is simple and unsexy. Distance beats exposure, time compounds it, and pulses may matter more than averages in narrow windows. Control the first 2, and the third fades into the noise. Now, what should a normal person actually do with that? Let's get practical without panic. Practical safeguards without panic. Let's make this simple and useful. No doom, just easy moves that cut the tiny risk even tinier while helping your biology handle normal stress. First, distance is king. Use speaker mode or a headset for calls. Wired earbuds are cheap, reliable, and don't need charging. If you like Bluetooth, fine. Power is low and bursts are short. Just keep the phone off your body. Pocket carry is the big one. If you must, flip the screen outward and put it in airplane mode when you're not using it. Better? Bag carry, jacket pocket, or desk. 10 inches buys you a massive drop in exposure, and you won't notice the difference after a week. Second, placement beats gadgets. At home, park your Wi-Fi router across the room, not on your nightstand or right next to the couch. Up on a shelf is great for signal and distance. If you can aim antennas sideways, not directly at your favorite chair. For laptops, use a table. Laps are for cats, not transmitters. And if you use a wireless charger, don't leave it right under where you sleep. Those coils talk at close range, then fall off fast with distance. Third, sleep like it matters, because it does. Your brain and mitochondria do a pile of repair at night. Create a quiet sleep zone. Ditch bedside chargers. Put the phone on a dresser or outside the room. Airplane mode after lights out is a tiny habit with a big upside. Fewer pings, fewer distractions, better sleep, lower baseline stress. If you're a parent, same rule for kids. Devices sleep outside bedrooms. Fourth, control the rhythm. Intermittent use is your friend. Set your phone to fetch email less often. Kill background app, refresh you don't need. Use Wi-Fi when available. It often runs at lower power than a phone climbing a weak cell tower. If your router supports schedules, let it nap at night. Not because the waves are scary, but because your brain is less late night scrolling, more deep sleep. Win-win. Fifth, stack the deck in your biology's favor. Antioxidant capacity is not a pill. It's a lifestyle. Move your body most days. Sleep walks, lifts, anything that raises your heart rate and drops it again. Eat actual food. Plants, protein, colorful stuff. Omega-3s, polyphenols, all the usual heroes that help your redox network handle life. Hydrate. Manage stress in any way that works for you. Breath work, friends, therapy, sunlight. When your baseline is good, small nudges, RF or otherwise, get swallowed by resilience. 6, be smart about fertility. If you're trying to conceive or you just care about your swimmers, don't park a hot phone next to them all day. Heat plus close-in radio plus low antioxidant reserve is not a combo you need. Keep the phone in a bag, not a front pocket. Avoid laptops straight on the lap for long stretches. Heat is the main villain there. These are boring, high-yield moves. 7th, skip the snake oil. There's a cottage industry of stickers, pendants, and harmonizers that claim to neutralize fields. If it doesn't change distance, duty cycle, or the device's transmit power, it's not doing what the label implies. Some shield cases can even make your phone work harder to reach the tower, raising power. If you're curious, borrow a basic RF meter from a library or a friend. Or use your phone's own field test mode to watch signal strength. Move things, watch numbers drop. That's your real shield, space and smarts. 8th, keep context in view. Millimeter wave 5G mostly sinks into skin. So if you're worried, don't glue the handset to one patch of skin for hours while streaming. Rotate hands. Use a stand. For wearables, weigh benefits. A heart rate monitor that gets you moving is probably a bigger health win than any tiny, uncertain RF effect. Set notification limits so it pings less. Let tech serve you, not poke you all day. 9th, mind the environment. In a car, let the phone use the car's Bluetooth or a cradle near the dash. Bodies block signal. Pockets make phones work harder. In elevators or weak signal spots, the phone ramps up. If you don't need it, then airplane mode until you're out. Tiny habit, easy savings. 10th, default to boring precautions for kids. Their habits stick. Teach speaker mode, desk parking during homework, and no devices on pillows. These are not fear rules. They're focus and sleep rules with a side of physics. Finally, keep your expectations sane. Current safety limits aim to prevent heating and give big cushions. Most everyday exposures sit far below those thresholds. The non-thermal debate is real science, not settled, and likely lives in small effects, narrow windows, and sensitive contexts. Your choices, distance, time, sleep, diet, push those contexts towards safety. Do the easy things, then get on with your life. That's the point. Practical, cheap, low-effort steps that help even if RF turns out to be a nothing burger. Because they also cut distractions, boost sleep, and improve resilience. That's the kind of hedge you can keep forever. Now let's zoom out and talk about what this actually means for how you live with tech without living in fear. What this really means for you. Let's be real. Your body is already running this delete and repair dance non-stop every minute. Millions of cells finish their shift, hand in the keys, and bow out clean. Others take a hit, patch up, and keep going. That's normal life. The question isn't, is apoptosis happening? It's, are we tilting the odds toward too much or too little at the wrong time? Modern life stacks small stresses. Some we can't dodge, some we absolutely can. Start with the big lever you control, baseline biology. If sleep is solid, food is real, and you move your body, your redox and repair systems run fast and clean. So a tiny extra nudge, heat, a harsh workday, a radio pulse, lands on cushions, not concrete. When sleep is wrecked and inflammation's high, those cushions thin out. Then small pokes feel bigger. This isn't a gadget problem, it's a margin problem. Next, the tech trade-offs. Faster networks move more data by slicing time into tighter pulses and leaning on higher frequencies. That gives us speed, but changes patterns. Standards focus on heating, with big safety margins. The open question sits in a narrow slice of possible non-thermal windows, where pulses could matter to sensitive cell types up close. That's not a plot twist. It's an engineering constraint. We can ask for better design, smarter power control, fewer idle pings, user settings that reduce chatter by default. Honestly, the loudest signal in your life probably isn't RF. It's light at night, doom scroll stress, and skip sleep. Blue-white screens crush melatonin. Late caffeine drags out cortisol. Those wreck repair far more than a router across the room. So fix the obvious first. Earlier screen off times, warmer light, a bedtime you keep. The repair machinery thanks you by buffering everything else. Could it be that some people are more sensitive? Sure. Biology isn't copy-paste. Sperm cells have tight antioxidant budgets. Neurons don't divide. Lose too many and function drops. Skin faces UV and pollution daily, then gets first dibs on millimeter waves. If you live in those edge cases, trying to conceive, recovering from a brain injury, working under harsh UV, pull the easy distance levers harder. Same rules, just stricter. What if you want certainty? You won't get it soon. Long-term, real-world RF studies are slow and messy. Tech keeps changing, measurement is hard, so you live with uncertainty like adults do. Make low-cost choices that help across the board. Speaker mode, bag carry, router not on the nightstand, sleep first, food with antioxidants that come in plants, not labels, exercise that makes you breathe and then calm down. Each habit shores up the same systems, the mitochondria, the redox buffers, the p53 checkpoints that decide repair or delete. Imagine a world where we treat tech literacy like nutrition, not fear. Literacy, you learn the basics. Distance beats exposure, pulses ride timing, and you set your defaults. You toggle off background app noise, you schedule quiet hours. You put the phone down when bars are terrible so it isn't blasting at max to reach a tower. You don't need a meter to do that. You need 1 week of trying. Plot twist. When you cut the chatter, you notice your brain works better. Fewer buzzes, fewer context switches, deeper sleep. You did it for RF, but the big win was time and attention. That's the theme here. You don't have to solve every debate to make your life better now. So where does this land on the central question? The delete command is real. It's there to keep you alive. Modern signals at everyday levels are unlikely to slam that key across healthy tissues. In edge cases and closed ranges, tiny nudges through oxidative stress are plausible, not proven. Your best move isn't panic, it's margins. Build a body that shrugs at small shocks. Set tech up so it whispers, not yells. Then get on with living. Now the verdict? Did modern frequencies hit delete? Let's answer it straight. Yes, there's a built-in delete key, apoptosis. It's real, it's precise, and it saves your life every day. The bridge from modern frequencies to that key is oxidative stress and calcium handling, mostly through mitochondria and P53. But is that bridge carrying heavy traffic in real life? Based on the best evidence so far, probably not for most people, most of the time. Here's what we know with confidence. Non-ionizing radio waves don't have the photon energy to snap DNA like X-rays do. That's physics. Safety limits are built to prevent heating with big buffers. When labs clamp temperature and dose carefully, many claimed effects vanish. That matters. In human skin cells hit with 3.5 gigahertz 5G, like signals up to 4 watts per kilogram, researchers didn't see oxidative stress or broken repair. Other work at 26.5 gigahertz also reported no DNA damage under tested conditions. That's a strong no in those models. Now the but. Some cell types look touchier under certain patterns. Sperm, neurons, some immune cells. In vitro boar sperm exposed to 5G band frequencies showed more DNA fragmentation in a frequency-dependent way. Animal studies sometimes find oxidative stress in brain, testis, or liver after weeks of intermittent exposure. Not always, not everywhere, but often enough to keep the question open. Modulation and duty cycles seem to matter more than raw power in a few of these. Signals, which tracks with biology listening to pulses, not averages. So, could everyday tech nudge apoptosis? In edge cases, yes. The plausible path is small. Fields bias calcium channels of hair, mitochondria work a bit harder. RAWS ticks up. If your antioxidant buffers are thin, poor sleep, chronic inflammation, heat right next to tissue, that bump could stabilize P53 a little longer and push a few vulnerable cells over the line. We're talking local and rare, not a system-wide delete storm. Fertility is the clearest practical angle. Close carry, plus warmth, plus time is a combo you can avoid with one habit change. What about big population signals? Tumor registries haven't shown a wave that tracks decades of rising phone use. Tower studies don't show consistent spikes, and far-field exposure is low. WHO's 2B's possibly carcinogenic label says, limited evidence, not at will. That bucket holds coffee some years. Keep that scale in mind. Where research points next: mitochondria, calcium windows, and modulation patterns. The experiments that will matter most tie a defined, realistic exposure to a reproducible early signal, calcium or mitochondrial potential, then to oxidative markers, then to P53 stabilization, then to caspase activation under tight thermal control. In people, the win would be clean biomarker studies with tracked handset power, not self-reported minutes. Your power is boring and real. Distance and time beat almost everything. Sleep, movement, and real food raise your buffers so small nudges disappear into noise. If a window exists in everyday life, you'll miss it more often with those two levers pulled. That's the honest verdict. The delete key exists, modern frequencies aren't mashing it. In closed, sensitive contexts, tiny nudges are plausible, not proven. You hold the finger on the key with how you carry, how you sleep, and how you live. By the time you finish this, your body has quietly deleted millions of cells and repaired millions more. That's not a crisis. That's maintenance. Modern signals aren't slamming the delete key, but closed pulsed exposures can be one more nudge on a crowded dashboard. Build margins, and the system wins. Want deeper dives on P53, mitochondria, and real device tests with meters and biomarkers? Subscribe, hit the bell, and share this with someone who needs calm, not clicks. Next up, we'll profile actual gadgets, map pulses, and track oxidative markers. You're not fragile, you're adaptable. Use that.